Okay, so influenza multiplication cycle. So even though influenza is part of Orthomyxoviridae family, and even though it shares a very similar name to Paramyxoviridae, its lifestyle is actually different from that of Rhabdoviridae and Paramyxoviridae. So for flu and Orthomyxoviridae in general, here's a virion particle. There are 8 native single-stranded RNA segments. Now, Orthomyxoviridae has a high hemagglutinin and neuraminidase antigens. So for attachment, flu uses hemagglutinin, and neuraminidase will be later on at the end of the cycle be used for viral release, and I'll tell you how that works later. So you have the hemagglutinin right here, and it attaches to sialic acid, and then you have the penetration. So just like all the other multiplication cycles, you attach, penetrate, uncoat, and then you transcribe and replicate. So you have the penetration here, and then you have the uncoating. The uncoating is right here. Now, it's a partial uncoating, so you still have the nucleocapsid with the 8 genome segments inside. And this is where, up until this point, we had learned about the other viruses and their hold on, wait. So polio multiplied in the cytoplasm, and so did Rhabdoviridae, and then the other two they could have taken place in the nucleus. I can't remember. But okay, anyways, so what makes flu special is that they don't make their own RNA primers for transcription, or they don't make it, unlike the previous 4 viruses. That's my point. And so it's essential for flu to go from the cytoplasm into the nucleus, because here it will steal the RNA primers from the cell using PB2, which is a flu protein. PB2 steals the cell RNA primers, and PB1 is the polymerase. And PB1 along with PA work together to make the polymerase complex, and then the native single-stranded RNA gets transcribed into positive single-stranded RNA, and so you have this primary transcription right here, right, and the virion-associated polymerase. So just like Rhabdoviridae and Paramyxoviridae, the flu has a virion-associated polymerase because it's native single-stranded RNA. So it needs that virion-associated polymerase to do its transcription and its replication. And again, the virion-associated polymerases are PA, PB1, PB2 together. So you have this, the single, these few copies of the positive single-strand RNA, which is basically, converted. It was made from the native single-strand. Now, because you're in the nucleus of the cell, since the flu is in the nucleus of the cell, it can transcribe these things and make messenger RNAs essentially, but it can't translate the proteins. So in order to translate the proteins, you have to go outside the cell, or go outside the nucleus and into the cytoplasm. So you see that here. And as from here, you have 8 different genome segments floating around in their positive single-stranded RNA form, and then you have that an initial cycle translation. And these proteins are made along with some of the other structural proteins, such as neuraminidase and hemagglutinin, and nucleocapsid, the polymerases. Now, from here, these proteins, NS1, M2, M1, NAP, PB1, PB2, PA, and nucleocapsid, they go back into the nucleus. Right, so you may be asking, why does it do this? What's the whole point? Well, you bring in more polymerase. So that you can perform secondary transcription. So while the virus was making these proteins, it took the positive single-strand RNA here. So, at the same time that it was making proteins the first time around, it was also replicating native single-stranded RNA. So you have all this. And so you have a bunch of native single-stranded RNA in a nucleus and a bunch of, structural and polymerase proteins coming in from the cytoplasm back into the nucleus. It allows you to make these progeny nucleocapsids. And from the progeny nucleocapsids, the nucleocapsids, like the premature virions can either go outside, or you can undergo secondary transcription. So you can make more positive single-stranded RNA from your native single-stranded RNA, and then you make from there, you have a bunch of positive single-stranded RNA. You can export those out and make more protein, right? And you can also replicate more native single-stranded RNA, and then you repeat the cycle over and over and over again, right? Until there's enough of the flu virus. So I said, let's do a different color. Let's do green. So here was the progeny nucleocapsid. I did say that you can go here, right? So this is just nucleocapsid transport, particle assembly. The neuraminidase and the hemagglutinin that is made during translation, they don't go back into the nucleus. They do not go back into the nucleus. They go through the ER into Golgi apparatus, basically for protein maturation, and they get stuck right here into the membrane before the budding happens, I guess. And when the budding does start to happen, the progeny nucleocapsid comes here, and you can see that you have the cell membrane. It's almost like it's ready to accept a virion nucleocapsid. So I said at the beginning of this video that hemagglutinin is used for attachment and neuraminidase is used for viral release. So how is neuraminidase used for viral release? Neuraminidase actually goes back. It cuts, or it cleaves up, chops up the hemagglutinin. It chops up the hemagglutinin sialic acid complex. And the reason for that is, if you can imagine the virion while it's budding, the new baby virion has a bunch of neuraminidase and and hemagglutinin, right? But there's still a bunch of sialic acid, that's still around on the cell. If you were the virus, would you want to bud out of the cell, and then because you have hemagglutinin, immediately lock onto the same cell that you came out of because of the sialic acid hemagglutinin complex? No, of course not, because the cell is used up. You don't want to go back into the cell that you came out of when that cell has no more resources. So the neuraminidase is important in cleaving those accidental hemagglutinin sialic acid complexes that result. So this is influenza multiplication cycle. Actually I forgot one more thing, so I'll use orange for this. This PB1F2 is another protein, one of the 11 proteins that are made by flu, And you see here it goes to mitochondria, and you may be wondering, why does that even matter? Essentially, what PB1F2 does is that it lowers our immune system. It antagonizes our immune system, so we can't fight flu as well. Okay, so this is the flu or Orthomyxoviridae multiplication cycle. I'll see you in the next one.